debris in the eyes, and because of keratin, the meibomian gland openings start to be clogged up by a biofilm, and it starts to be keratinized, which is what leads to lid inflammation and dryness over time. So what we are saying now is that this biofilm buildup on the lids causing meibomian gland dysfunction is equal to teeth plaque. And if we can start training our patients to actually look after their eyelids like they do their teeth, wash every day, brush every day, a little bit of heat, keep those lids clean on a daily basis, we are probably going to see less lid issues, less meibomian gland dysfunction, and less dryness problems in the long term. And then this is something I just wanted to share with you guys just on the note of disease. So I had a colleague that said to me, it's not rare if it's in your chair. And this is something that none of us ever really want to see. But this little boy is Stevens-Johnson's. He has Stevens-Johnson's disease. And the problem with Stevens-Johnson's disease, it creates this scarring and severe kind of scarring of the lid, in this case, the lid margin, which created this entropion. And by the time he got to me, the entropion and the eyelid or the eyelashes had created such bad scratching on the corneal surface that the one eye had already perforated. And this is what he looked like when he got to me. So the left eye had a complete full thickness perforation because of the actual rubbing of the eyelashes. The right eye was down to about 180 micron on that central cornea. And just because the eyelashes wasn't pulled out and he had this entropion that formed. And in this case, he has absolutely zero aqueous production. There's no tears in that eye whatsoever. So this is dry eye disease. And with him, it's very hard to manage. We are in management with him at the moment. The right eye is doing quite well. Obviously, there's nothing to do with the left, but yeah, this is how bad dry eye can get. And this is the stuff that we should really be looking out for. Okay, and then just as a bonus, I thought I wanted to share this with you guys. So I have structured a grading ebook. So the grading ebook was there to be able to make sure that the way that we actually grade our tests is universal and standardized, so that when we all start talking across countries about this dry eye, we start data collecting, we make sure that we're all doing it on the same level. The Efron scales and all of that is available. But what I've done is I've put it all together in terms of these tests, made sure that all the norms for each test is there. I've also just popped the link within the chat box. So please, you guys are very, very welcome to go and download your free copy. It's free. You guys are very welcome to go do that. It's really helpful. It's an ebook, so you can have it on your iPads, telephones, computers at work, whatever suits you best. Right. And then that's me. So yeah, thank you guys for the opportunity. I would love to know if there's any questions.
Thank you so much for your lovely presentation. It was very useful. For me, at least, it was perfect. So guys, no rose bengal anymore. And for Scheimer test, please apply anesthetic, okay? We don't want reflex tears. And one question from me first. What suggestion would you give to parents to just prevent their child to get dry eye?
I think the main thing is that this whole lid care process needs to be started, from young. I think parents need to understand that, and like I said, it needs to be part of your daily routine. Like you brush your teeth, you're going to wash your eyes. I do think that is in itself going to play a huge role in eliminating a lot of issues going forward. I must be honest with you, this meibomian gland atrophy thing has kind of fallen on my desk in the last 3 or 4 weeks. It's brought up a lot of questions in my mind about why this is happening. And I don't have answers for that yet. So I'm very excited to try and find some answers, because there needs to be another factor at play other than just the the lid thing. There has to be something else. And as a mum, as the person who has the child who is struggling with this, I'm very much, wanting to look into why. But I think for now, and research shows that lid hygiene, lid wiping, and the warm compresses really does help to keep the meibomian gland structure functioning well. So let's go with that until we have any other solution.
More research, right? And thank you for that. And there is a couple of questions for you. Manish is asking, what about lip view, or which is better for gland imaging, lip view or topography?
To be honest with you, I'm actually just sitting here Googling it. I don't know LipiView very well. I see it's an interferometer, so I've actually never come across it from where I'm at. So I can't have an honest opinion. I can have a look into it for you. So as long as it can do what you want it to do, I don't have a preference in terms of equipment. So I like to look for the thing that can do as much of what I need in one type of machine, which the topographer has always given me, because you can do your topography, but you can also do all your ocular surface stuff as well. So I'll have a look into the LipiView. I don't know it, so sorry, I can't give you a clear answer on that.
Yeah, they can just contact you through your email, if you can just say your email address. So
what I'll do is I will type it in in this chat box if anyone wants me.
Thank you. I appreciate that. Are you ready for another question?
Yep, go for it.
One of our participants is asking, hi ma'am, my question is, can meibomian gland dysfunction increase from acute to chronic as we get older and older if untreated?
That's a very good question. Yeah, the answer is yes, for sure. Meibomian gland dysfunction or meibomian gland atrophy, depending on where you are in the system, is something that will continue to get worse if not treated. So the good news about meibomian gland dysfunction is it doesn't take a week or two to atrophy altogether. According to what we see, is it takes quite a long period of time before it actually completely stenoses and dries out. What we are trying to do is trying to shift the focus from managing meibomian gland dysfunction to preventing meibomian gland dysfunction altogether. And if we can actually do a lid management from a young age, then we shouldn't have, or we shouldn't see any meibomian gland dysfunction going forward. And if people can continue their home care chronically, then surely this thing should not get worse. So yes, if untreated, if someone doesn't clean their lids, if there is inflammation in the glands, it will continue to get worse. It will atrophy altogether, and the patient will be sitting at 70 years of age without any meibomian glands, which really is a problem in terms of comfort.
Yes, right. A question just came in my mind is like, if any kid come to your clinic and they are just suffering from eye fatigue or blur vision, it's not, we should just not jump into refraction. I think first we should just do dry eye assessment. Maybe it's nothing to do with the refraction or refractive error.
And you know what I found? Kids are incredibly tolerant. They're incredibly tolerant of a lot of things. You will find a child in your office with a -2 and he doesn't even know he can't see. The same thing happens with dry eye. They are so tolerant of what they experience because they don't firstly know how to verbalize it well, and secondly they just kind of deal with it. And I've seen it in my own daughter. I mean, she has this chronic allergic conjunctivitis. She never rubs her eyes. Her eyes are never red. They are never puffy. And then I looked at it and she's got this papillae all over. And it just kind of changes our perspective in terms of how we deal with our kids and how we deal with the pediatrics in our room, because blurry vision is very often ocular surface related and not script related. So now we're giving kids plus 50s with a blue control because it's computer overuse, but we're not having the discussion of blink more, wash your eyelids, you have a dryness issue. We're not having this discussion. We're just giving them blue control. And if the blue control works, it helps, but it doesn't fix the whole problem. So I agree with you, Sumaya. I think very much so. We need to look at the ocular surface before we look at the refraction.
Yeah, thank you. An interesting question from Komal is, what lifestyle changes we can suggest to our patient?
Yeah, that's a difficult one. And you know what? I find that difficult is because it's very hard to change someone's lifestyle. It's very hard to tell a patient what he should and shouldn't do. And I'm equally guilty. I mean, I can sit in front of my computer for like 7, 8, 9, 10 hours. If I was such a coffee addict, I would probably not even get up. So like for me, I get up and make coffee every so often, So I've taken a little bit of a different approach to managing patients in terms of how we speak to them. We try and fit our idea of what should happen into their lifestyle. So your idea, okay, good, you need to not spend so much time in front of the computer, or you need to have dryness related management. Okay, so how can we make this part of your lifestyle so that you actually do what I ask you to do? Because if you take it out of their lifestyle, you expect them to change their lifestyle, it's not going to happen. They're just not going to do anything. So you want to try and meet them halfway. So for the guy who sits in front of the computer, you want to say to him, okay, do you have a window? Do me a favor and just count some leaves on the tree every hour, just to get him to defocus away from his computer. Because if you're going to say to him, stop working on your computer, it's just not going to happen. The same goes for dry eye. If you're going to ask them to do lid management, now you say to this, and I know my husband has it. So you say to this gentleman, please take these round cotton pads and stand in front of the basin. And I mean, really, he's not going to do that. So get him to do it in the shower with the face cloth and just make it part of his routine. Otherwise, it just doesn't get done. And the moment he starts having improvement, then you can add or change a few things. But try and fit it into your patient. Don't try and fit your patient into your idea.
Exactly. Unfortunately, social media and cell phone looking is part of our routine life, and we should get used to just come across many dry eye patients. Actually, we should go and just grab them and just teach them. I think there should be lots of vision screening and consultant at school or even kindergarten. Yeah.
Yep.
Okay, there is another question from you. How effective is the meibomian gland stimulation therapy in decreasing the meibomian gland atrophy, mainly in moderate degree of meibomian gland dysfunction?
Okay, so the biggest challenge that we've always seen is that once the gland is atrophied, it's almost impossible to regain it. If there's dysfunction in the sense that the gland is not expressing, but the gland still exists, or the expression is not fluid but pasty, then the recovery is incredible. Just by doing some heat treatment and in-office therapy specifically, maybe with some gland probing or whatever is necessary, we actually get almost an 80% recovery of gland function, but that is in an existing gland. So the gland has to be there. Once it atrophies, we have not been able to regain an atrophied gland, which is why this is so important, because once it atrophies, you can't regain it. So, but in terms of your question, gland stimulation therapy, whichever way you decide to go with that, is highly, highly effective. I have had incredible results with it, and I've taken patients off of drops completely just by doing in-office gland therapy.
Right. Thank you so much, Mariki. It was very useful. Good talking to you. And
Thank
I
you,
think
guys.
we'll have some further presentation from you, like the management of dry eye, which I'm very interested to hear from you.
Thanks for having me, and I would be absolutely glad to do that.
Thank you so much. All right. Now I'm going to introduce the next speaker, which is Miss Mega Chatterjee Mega is going to talk about the Drosophila. Actually, it's very interesting topic. Hello, Mega.
Hi, hi, Sumaya. Hi.
How are you? Okay, how the
Yeah, I'm all
PhD
good.
going? I think you are just pursuing your PhD in University of Delhi.
Yeah. So I'm in my final year of PhD and just about to. So my research work is complete as far as my PhD goes. So it is done, and now I'm going to submit my thesis. Hopefully, it goes well soon.
I should have called you Dr. Mega.
No, not yet. Hopefully soon. Hopefully
I,
soon.
of course, of course. All right, this is you and the stage. Carry on, we are listening.
Okay, so let me share my screen first. All right. So let me know if my screen is visible to everyone.
Yes, that's very clear.
Thank you. Thank you. So yeah, first of all, hello everybody, and we just heard some great talks from the presenters. So thumbs up to the presenters, and also to Vision Science Academy team for organizing such a great event. So thumbs up to all of you. So okay, so now what I'm going to talk about is I'm going to take you all a little away from the clinic and into the laboratory, and for the next 15 to 20 minutes, and we'll be focusing on the eye of a tiny little creature, which is Drosophila. So, uh, so Drosophila is commonly known as fruit flies. They are also known as vinegar flies, wine flies. Now, it is clear from their names, they are generally spotted over overripe and rotten fruits. And so, within this genus Drosophila, there are over 1,500 species identified till date. However, Drosophila melanogaster specifically is used in biological research. Majorly, it is used in biological research. So as you can see in my slide, on the right-hand side, it is the male fly, and in the left-hand side, it is the female fly. So sexual dimorphism is very much clear, phenotypically as well. As we can see that the female flies are slightly larger in size than the male flies, and at the posterior end of the male flies, you can see black patches over the abdomen. However, they are not too large. Also, it is not visible in the picture, but under the microscope, on the forelegs, there are sex combs present in the males, which are not present in the females. So they are very much identifiable. Now, why Drosophila acts as a model system? And by saying model, how they can do it is approximately 60% of Drosophila and human gene, they actually share homology. And also 75% of the human disease causing genes, they are present in the Drosophila genome. Secondly, and most importantly, they have a very brief generation time. If we look into the life cycle of Drosophila, we can see that the adult female, it lays eggs, and on the following day, the first instar emerges. And it goes on from first instar to second instar, then to third instar. And the third instar larvae is the final stage of larva. And what happens is when you culture in a container, within the food, first instar till the third instar, they are within the food only. And after that, during the late third instar stage, they move up from the food and they go onto the walls of the container and they wander there. And then after 2 to 3 days, they pupate. After pupation, they go to adult stage. So within 11 to 12 days, actually, we get another generation adults. And when after 8 to 10 hours of emergence, the fly is again sexually mature and it can mate and then lay eggs. So within a span of 11 to 12 days, we are actually getting next generation. So it becomes very a speedy procedure to get generation after generation, and then validate our experiments and everything, do things about them. Also, a larger sample size is a great advantage for Drosophila, as one female can lay approximately 200 eggs in in its lifetime. So, we get a great amount of sample size, which is very important to infer results, because greater the sample sizes, we know the results are more reliable that way. So also the maintenance of these cultures are very economical. They do not need very high end scientific equipments. Just the maintain the temperature and you're good to go with the cultures. Also, they are very powerful genetic tools, and by saying that, in Drosophila, it has 3 chromosomes versus 23 chromosomes in humans. So you know that it is very easy to handle, and genetic engineering becomes very easy to do, and also the genome is well sequenced. So we can get very well. Now, coming to Drosophila I. Now, as I said that there are larval stages and there are adult stages. So we can work on both of them. In larval stage, we have eye discs. That is, each adult structure, they are present as monolayer epithelial tissues within the body of the larva, whether it is eyes, legs, wings, everything is present as an epithelial tissue. So if we look into the Drosophila eye, so it is present as an eye disc, and they contain photoreceptor neurons arranged in approximately 20 to 22 rows, and they are beautifully patterned in the eye disc. Now, when this eye disc proliferates and differentiates, it gives rise to the beautiful brick red adult eye. Now, these are all microscopic pictures, and just simple, bright microscope does the job well. Now, in the adult eye, we can see that the complete surface area of the eye, of the Drosophila eye, is covered by small rhomboid structures, and they are approximately 800 in number. Females exceeds a bit from the males. So approximately 800. Now, each ommatidia has 8 photoreceptor neurons, 4 cone cells, and 2 primary pigment cells. So these pigment cells actually give the brick red appearance. Now, if we look into the structure of each ommatidia, you can see that there are the rhabdomeres. Rhabdomeres is actually another name for the photoreceptor neurons I just mentioned. So there are 8 rhabdomeres. There are 2 pigment cells around it. There's a facet lens. And now the top view can give us this arrangement. Now you can see here are 7 rhabdomeres which are present. However, the 8th one is below the 7th one. So the 8th one is usually not visible if we get a top view of it. Now, why am I saying that Drosophila can actually model human diseases? Now, basically, what happens is many human diseases are genetic in nature. So what happens is Drosophila has a beautiful system of US GAL4. Now, what a GAL4 is? GAL4 is a yeast transcriptional activator. And when it is associated with a tissue-specific enhancer, it actually gets expressed in a tissue-specific manner. And and it when attaches to an upstream activator C gene of interest. So whatever our gene of interest is, whether it is a human gene inserted into the Drosophila, whether it is any mutation we are getting into, so that gene would get expressed within the fly, and we can get the transgene expressed only in that tissue which we want. So ubiquitous expression is not needed. So there are various diseases which are modeled in flies, and disease mechanisms are unraveled using this fly model. And as I said that, when we go into clinical trials, we cannot have that much sample size, but we can have up to thousands of sample size when we work on Drosophila. So you can now know the power of this model organism. So several developmental disorders, neurodegenerative diseases, even cancers has been modeled in this disease, and many drug discoveries has been done through this. As I said, that we can express any foreign gene in specific tissues. Now, since we are talking about eye as a platform to study, so we can see here, so there's a protein called GFP. Now, GFP is not any endogenous protein of Drosophila. It is a green fluorescent protein which fluoresces and gives away the green fluorescent light when illuminated. So we can see throughout the developmental stages that GMR GAL4. Now, as I said, that GAL4 is Galfor can be expressed in a very tissue-specific manner. So GMR Galfor is actually an eye-specific driver. So GMR Galfor has driven the expression of GFP in only the eyes. So we can see first instar till second, third, and also pupa and adult flies. So we can see that GFP is getting expressed only in the eye. So in this manner, we can also express several human genes only in the eye of Drosophila. Now, coming to how can we actually visualize the toxicity. Now, it is very interesting to know that any toxicity that happens in the whole body of humans, it can be illustrated using only the eye as a platform. So we can see now here, as you can see, the control eye in the extreme left panel. Now, then we inserted one disease protein. Now, it can be any disease protein of a human that causes cell death or cytotoxicity, as we say it. So we can see high depigmentations happening here. Further, if we increase the levels of expression of that, or we also induce any other disease proteins, we can see necrotic lesions and some of the depigmentation happening here. So these are all just, you do not need any high-end equipment. The beauty of this model system is you can just see it under a microscope and you can visualize what all toxicity is happening around. We can see here that retinal degeneration is happening due to insertion of any disease gene. Now, this is a very interesting picture. What we did is we dissected the eye, we kept it on a slide, and illuminated the eye from beneath the slide. So what happens is, from each ommatidia, you can, if you look closely, you can count the number of photoreceptors here. So these are 7 in number. As I said, on the top view, you can only visualize 7 photoreceptor neurons. And then in the disease model, you can see a lot of loss of photoreceptor neurons here. So this is how toxicity can be visualized. Now, there's many things into it, but I cannot compile it in a 15-minute talk. Now, if we say that we want to visualize a protein, now we know there are many neurodegenerative diseases, whether it is Alzheimer's or Huntington, they are associated with protein aggregates. You know, the causative proteins, they get aggregated into the neurons. So, you know, exactly like that, if we looked into the photoreceptor neurons of flies, so these are actually larval eye antennal discs, which I earlier mentioned. So we can see we have tagged with antibodies specific to disease-causing protein, and we can see several aggregates happening around here. So the red panel is actually nothing, but it is tagging the photoreceptor neurons, and the green panel is tagging the protein aggregates. And this is the merged panel. We can see now, if we increase the zoom on optical zoom, or if we increase the the lens power, we can see a highly magnified image, and we can actually know whether the protein is within the nucleus or whether it is in cytoplasm, and what all is happening around the protein. So proteotoxicity can very well be mirrored here. So this is what I wanted to say about the Drosophila eye. So the take-home message is toxicity associated with the whole human body. It can be illustrated in the tiny fly eye. And so you can all imagine how magical creation of a nature it is. And also the main point is, it is very easy to do. We can have various advantages associated with this model. So yeah, so it is a great platform to study our human diseases. And not only, I would like to add that not only the molecular mechanisms are unraveled, but various drug discoveries also happen here, so that you can feed a drug and then look out what all impact it is actually giving out. So thank you, and I welcome questions.
Thank you so much, Megha. It was really interesting. Drosophila, thanks to all the Drosophilas. And I just want to know how much, like how long is their lifespan?
See, the lifespan actually varies, but in general, if I would be saying, it is approximately, it varies around 60 to 80 days, depending on the species or depending on what line it is. But it varies around 60 to 80 days, usually.
Yeah. So basically, all the photoreceptors are located in the center of their eyes,
Yeah.
as I.
Yeah.
Oh, that's
From
amazing.
each ommatidia. Yeah.
That's very amazing.
Thank you. Thank you.
Yeah. Oh, we've got a question from you. All right. What are the possible relation in regards to Drosophila eye in treating macular degeneration and other human degenerative diseases? Um, I'm not
See,
sure if you live on. Okay, if you can answer, would be very great.
what I got from the question, I would try to answer that, like in neurodegenerative diseases associated with humans, if I would say, so there's a lot of degeneration happening in the brain. I'm not saying that the degeneration happening to the human eye, but what I'm saying is all the neurons which are getting affected, we can actually mirror it in Drosophila eye. So we know that cytotoxicity, what is happening in the brain of the humans, it is actually can be visualized in Drosophila eye, and we can play around with many drug treatments, or we can unravel many. So that's what a model does. It doesn't replicate the disease, but using, and I'm talking only using the eyes. However, if we express the protein all over the all the neurons, definitely many symptoms of neurodegenerative diseases of humans actually get mirrors in the eyes. That is, they get mobility issues, they get cognitive issues and psychiatric. I mean it is highly mirrored. But if we are talking only about the eye, we can say at the cellular level what all cytotoxicity is associated with it.
Yes, thank you so much.
Yeah.
And the last question from you is, what about the color of Drosophila's eye, brown or white? Can you please discuss about the eye color with the genetic studies in ophthalmologists?
Yeah, actually, what happens is, so many mutations are associated with this eye color. The wild type. If we talk about the wild type, it is brick red in color. However, there are many mutations. There is sepia mutation, there is white eye mutation. So these mutations, what does is white eye mutation. We have a complete white eye. In sepia mutation, it has complete brown eyes. It even, there is a mutation of eye which is bar, where a slit like structure just remains, and the complete eye just is not there. So there are several mutations that have been discovered through Drosophila eye. So it is like that.
Yes, I hope you just got the answer.
I
Thank you
hope
so
so.
much for just
Thank
participating
you.
in
Thank you.
the webinar. Thank you.
Thank you. It was lovely participating here. Thank you so much.
Okay, guys, I hope you enjoyed till now. Now I'm just quickly introducing the next speaker as we are running out of the time. Dr. Mukesh Jihai is with us. He is going to talk about optometry in Nepal. Mukesh completed the Bachelor of Optometry. Hi, Mukesh, and Vision Science from University of Technology in 2009. He worked as an optometrist for 1 year in Medical College in Nepal, and he has done his OD from Philippines and also master degree from my favorite college, Bharati Vidyapet University in Pune, which I've done my study from there. Very welcome, Mukesh, today to our webinar. We don't have your voice.
Now, hello.
Yes. Very welcome.
Hi,
Hi.
Sumaya, how are you?
I'm fine.
Great. Uh, Let me share my screen first.
All right.
Okay. Can you? Can you see my screen?
Yes, we just see your background screen.
Background screen. The slide now?
Not yet.
Not yet. Okay. 1 second. now?
Uh,
Again,
it's
why my
loading.
slides is not showing? Only folders only.
Okay, you just open your PowerPoint presentation. Okay, when you open that, then you have to just press the share screen in
I
your
can't
Zoom
open.
page. Mhm.
Uh, stop sharing. Just wait. Again, last time.
Oh, no problem. Take your time.
Uh, oh, yes.
Yes, now it's perfect.
Now it's
You
fine.
can. Yes, of course.
Hello. Now you can see.
Yes, Dr.
see
Mukesh. Now, yes.
Am I audible to all?
Yes, we are very audible. We are
Okay,
just
fine.
eagerly waiting to hear you.
So can I start now?
Yes, please.
Okay, fine. So namaste from Nepal. I am Mukesh Kumar Jha, OD, I'm Optum, now working in Patan Academy of Health Science. I'm also Joint Secretary of Nepal Association of Optometrists now. So today I'm going to talk about optometry practice in Nepal, how it started, and current scenario, how what is the role of Nepal Optometry Association for the development of optometry in the country like Nepal. Okay. So optometry educations in Nepal started, first graduate optometry program in Nepal started in 1998 from the Institute of Medicine BP Koirala Lines Center for Ophthalmic Studies in association with School of Optometry, University of Auckland. Initially, it was 3-year course and upgraded to 4 years in 2003. Subsequently, strengthened by World Council of Optometry, WCO, Charisov, Blind and Mission. So here, students are has very extensive clinical exposure in ophthalmology as well. Fewer opportunities for optometrists at the time, mostly recruited on personal recommendation, not
Dr.
much.
Sorry to just interrupt you. Can you please use full screen?
Full screen. Okay.
So much.
It's okay now.
Yes, that's much better now.
It's okay
Thank
now.
you.
Okay, fine. Thank you. Okay. So most of the graduates from Institute of Medicine looked for the opportunity outside the country. First batch was graduated in 2001. Before it was like 4 to 6 total seats, and now it's 10 seats. This was only optometry school until 2017 in the country. Nepalese Association of Optometrists formulated in 2004, and in 2005, optometrists got registered in Nepal Health Professional Council. As we don't have separate optometry council in the country, so we do registration in Nepal Health Professional Council. The Code of Ethics defined the clinical extent of optometry practice and created its legal basis in 2014. The registration of Code of Ethics with the government body made the course popular among students, and many of them went abroad, mostly India, to get optometry degree. Few of them got Doctor of Optometry from Philippines. Graduated number of optometrists started to increase significantly, and people and eye hospital become aware of the important cadre in the field of eye care. So before it was the only like before 2017 there is only one school in Nepal for bachelor in optometry and vision science. Now after in 2018 the second graduate optometry program in Nepal started. So one university, National Academy of Medical Science, it's short form NAMS. So total 40 seats in this university, and in Pokhara University total 15 seats started bachelor in optometry and vision science degree. Subsequently, Pokhara University total 12 seats started masters in optometry program. So this is the first time in 2018 Pokhara University or any other university only Pokhara University started masters in optometry. There was no masters in optometry before. Only bachelor in optometry started in 1998. Okay, the number of students vying for optometry is increasing. Nepalese Association of Optometrists since 2004, it started in 2004. So got registered at A level for B optometry and a specialization certificate for master's degree in the in Health Professional Council. Code of Ethics formulated in 2014 by advocacy from Nepal Optometry Associations and senior optometrists. In 2014, member of World Council of Optometry and Asia Pacific Congress since 2008, conducted school screening beneficial in providing service to get populations to general populations and more optometrists more familiar to the society, conducted awareness and provided eye care service to general people and school children in earthquake affected area. So these are the now work from 2004. Conduct first all Nepal Optometry Conference in 2018, conducted CEO in regular interval, supported optometrists and optometry students to participate international conference, advocated for need of optometrists in government integrated eye care service. Okay, integration of eye care within the basic health system. So we are doing work on this. Vacancy for optometrists at government level. Our associations has been advocating for optometrist position at municipal level and had been talked with the newly formed eye, ENT and dental department within the Department of Health and Population. Formation of Optical Act. So now from 2-3 years we are trying for the Optical Act. Now is also advocating for Optical Act, which would legalize the dispensing ophthalmic prescription to control the quality and price in benefit of people and professions. So Nepal can become the first nation in South Asia to introduce this act. This could further lead to Optometry Council, as we don't have our own Optometry Council, only Nepal Health Professional Council, where we registered there. Okay. Regulations with Nepal Health Professional Council, provision of a skill test and written examination before practicing license being issued from there. So now, current scenario in Nepal for optometrists are, optometry practice in Nepal is very broad. We are bound to practice as a mini ophthalmologist practice. So as a mini ophthalmologist practice, meaning because there is lack of awareness to the populations about the optometrists, even they want everything from single optoms. So we do all except surgeries, and our code of ethics also allows. So that's why we call as eye physician. We take detailed history, check vision, do refraction, perform slit lamp examination, and finally fundoscopy as a daily basis. We use drugs to help in diagnosis of ocular disease and prescribe drugs to treat those diseases. We also remove corneal foreign body. Few of us practice vision therapy, ocular prosthesis, specialty contact lens, and low vision. Kindly Google search optometry code of ethics in Nepal. So for our code of ethics. So number of optometrists increasing nowadays, number of vacancies appearing now and then. So it's still a lot of unexplored area, because as uh, like we have less practice of vision therapy, uh, this low vision, all necessity to develop subspecialty within the field of optometry. Uh, let's make our voice stronger, and let's join the fraternity of now. Thank you. Thank you for the presence. And thank you, Vision Science Academy, for giving me opportunity to for the talk. And thank you all once again.
Thank you, Dr. Mukesh. I can imagine how hard work you guys have done to just register optometry to the world, which unfortunately, ophthalmologist doesn't want us to have private clinic. And I'm living in Turkey, and here optometrist and optometry subject doesn't exist. So I hope one day, one day also it happened here.
Okay.
So how is contact lens practice over there? Are there people just well educated to use contact lenses?
Okay. So there are 3-4 cities like Kathmandu, Pokhara, Biratnagar, and two, three more cities where contact lens practice is very high compared to other places. Uh, like for soft contact lens, all are practicing that, but for the specialty contact lens, in only in hospital based practice, especially in Kathmandu, Pokhara, and Biratnagar, are hospital based practice. Uh, for soft lenses, all over the country, all optoms are practicing here. So for specialty, only hospital based, and only few, one or two, I can say, they are practicing specialty lenses outside, like private practice.
Yes, I hope you get next conference coming very soon, and we will all from VSA will be there for sure, because Nepal is a beautiful country to see.
Thank you.
There is a question from you. Well done, great historical development over the years. What are the possible collaboration options with international bodies in expanding opportunities, especially in public health area?
Okay. So our optometry friends, they have also done in like masters in public health. So they are and most of the optometrists who are graduate from Nepal Institute of Medicine, they mostly they are abroad, and they are collaborating from different organizations for Nepal Optometry Associations, or at least Nepalese optometrists to grow up. So definitely we are looking for collaborations, or anything, any organizations want to collaborate with us, or even we can communicate with them.
Yes, thank you.
Okay.
Another question from you is, can ethics of optometry of Nepal allow to add prefix doctor for optometrists?
Okay, so uh, now we are registering, like in Nepal Optometry, Nepal Health Professional Council. So council doesn't allow us to write doctor, or they haven't given the permission for DR, but certain hospitals, those who have done ODs, doctor in optometry, they allow them for to write the preface doctors. But what I think, up to now, in our country, like those who have done doctor in optometry or OD, they should write DR prefix, but other are not allowed. But for overall, we haven't like legalized to write DR in our country.
Yes, thank you so much, Dr. Mukesh, for you to being here and presenting us, and we got some broad view about
Yes.
optometry practice in Nepal.
Yes. Thank you. Thank you. Thank you.
Thank you so much. All right. Thank you, everyone, for joining us today. Please follow us on visionscienceacademy.org website for upcoming webinars, news, and plenty more advanced and recent eye-related subjects. I encourage you to be well, stay safe, and protect your eyes. Thank you so much.